What happened? I don't know what happened to my PowerPoint. I think what happened is when I was doing the screen share, I accidentally exited out of the thing. So let me go back and reopen that lecture. Okay, can everybody see the slide for the introduction to viruses? Make sure I have it right. Can everybody see the slide presentation?
Yeah.
Okay,
Yes.
okay, thank you. All right, so we're going to talk about viruses. I have a few more people to let in. Hold on. Alexander. All right, so Pasteur developed a vaccine for rabies, and he's the one who coined the term virus, which is Latin for poison. Ivanoski and Bereznik discovered that tobacco disease was caused by a virus, and Loeffler and Frosch foot and mouth disease is also discovered it was caused by a virus. So
Is
how
the
did they,
PowerPoint supposed to be up? I don't see it.
other people said they see it, or
There is no PowerPoint. We just see your file.
No, I can't see it.
No, his cousin had it and was leasing it.
It's in the what? In the
whatnot,
chat.
so I guess he got it.
It's chapters. I don't see the pictures of it or the words.
Right.
But he told
okay,
me,
what I'm gonna do is stop the share for a minute and go back. I don't know why some people are seeing it, some aren't. So do you see the PowerPoint for viruses now? It should say filterable virus.
Yes, I see it now.
Okay, sorry, maybe I clicked on it but didn't share it. So back when viruses first discovered, they didn't have electron microscopes and all the tools we have today. So what they did have is they developed these porcelain filters to trap bacteria. So they had very, very small pore sizes. And they got a liquid that would come through a cell-free filtered fluid, and they found that fluid was still infectious. So it had something in it, even though they couldn't see it. And this proved that there was something smaller than bacteria that could cause disease. So viruses have some unique properties. We don't consider them as cellular organisms. We rather think of them as infectious particles. And they are considered either active or inactive, but we don't consider them necessarily as being either alive or dead. They are obligate intracellular parasites. They cannot multiply unless they evade a specific host cell. They must instruct the genetic metabolic machinery of their host cell to make new viruses. So they basically, all the things we learned about DNA replication, protein synthesis, that's in your cell, the viruses hijack all that, use that to make new viruses. So as I said, they're not cells, they're obligate intracellular parasites. They can,
He's supposed to step up here, try it.
I'll call him.
okay. They can infect just about anything, bacteria, protozoan, fungi, algae, plants, and of course, animals. They do not independently fulfill characteristics of life, which is being a single cell organism with a cytoplasm and a membrane. They do not have those characteristics. They are inactive macromolecules outside the host cell and only active once they're inside a host cell. They have a pretty basic structure. They have a protein shell, or what we call a capsid, that surrounds their nucleic acid. They are ubiquitous in nature, and they have a major impact on development of biological life. They are ultra microscopic in size, ranging from about 200 to 1,000 nanometers. In other words, they're about the size of ribosomes inside cells.
Professor,
Yes.
I get confused when you say that something is ubiquitous. What do you mean by that? I don't really understand what that means.
Sorry, that means it's everywhere. Ubiquitous means it's everywhere. Like if I said our atmosphere is ubiquitous, that means it's everywhere. Is that clear? Okay. Viruses, their genetic material can be either DNA or RNA, but not both. They can have double-stranded DNA, but they can also, some of them can have single-stranded DNA. They can have single-stranded RNA or double-stranded RNA. Most DNA viruses are double-stranded, and most RNA viruses are single-stranded. On their surfaces, they have molecules that determine their specificity for attachment to a host cell. Multiply by taking control of the host genetic material and regulating the synthesis and assembly of new viruses. They lack enzymes for most metabolic processes, so they don't carry out glycolysis or aerobic respiration. They lack machinery for synthesizing their own proteins. So they have to use whatever their host cell has. This shows you trying to see how sorry, I wanted to enlarge that. Okay, so the viral size range, you can see up here, they're smaller than the average bacterium. Electron microscopes are required to see them, cannot see them with a regular light microscope. This shows you a comparison of some various viruses. I was hoping I could enlarge that picture, but you can look at it on your own. This shows you from an E. coli. And sorry, I can't even read what the smallest one down here is, but
What you want to do is escape out of it, and I'll show you how to enlarge it.
okay.
Hit the escape button, click that, click it, the picture. Now,
Oh, right.
yeah, and then take it and drag it. There you go.
Okay, that's what I was trying to do before and I couldn't get it. All right, so might as well just look at it. So you can see a hemoglobin molecule, yellow fever virus, quite small, polio, the viruses that infect bacteria. The bacteriophages are very small. The adenovirus, which a modified adenovirus is used in the Johnson & Johnson vaccine. influenza virus, HIV virus. This is a streptococcus bacteria, and E. coli bacteria, rickettsia, which is an obligate intracellular parasite, but it's a bacteria that are quite small. There's some larger viruses. So methods of viewing viruses. These are various electron micrographs. They've been colorized by a computer because at that small size and using electrons, you do not see visible light. Viruses bear no resemblance to cells. Everything that we think of as living, plants, animals, fungi, bacteria, all the animals, they are all cellular. The smallest unit is a cell, all cellular-based life forms. They have no protein synthesizing machinery, need only those parts required to invade and control a host cell. They obviously have to have the genetic material, and they are surrounded by protein capsids. Sometimes they have an external cell membrane, but that is not something they make. They take it from the host cell that they infect. So the capsid is what protects their nucleic acid. It is made up of proteins. The nucleic acid, along with the capsid, is referred to as a nucleic capsid. An envelope, not all viruses have an envelope. The envelope viruses is a modified piece of the host cell membrane that the virus takes when it leaves the cell. Some viruses are what we call naked viruses. They don't have the envelope. They consist only of the nucleocapsid. What they do, either on their protein capsid or on the envelope membrane, they put protein spikes. They project from either that nucleocapsid, which is the protein surrounding the genetic material, or in the envelope. These protein spikes allow viruses to dock with cells and to enter them. A virion is a fully formed virus that is able to establish infection in a host. So these are two viruses for comparison. So naked virus would have its genetic material surrounded by this protein capsid. Has usually a very geometric structure, and it will have some protein spikes to attach to a host cell. An envelope virus is still going to have the protein capsid surrounding the genetic material, but then they're going to have a membrane they take from the host and they modify that. They put protein spikes in the membrane that allows them to attach and penetrate into host cells. The viral capsid is the protective outer shell. It's made of proteins called capsomeres, identical protein subunits that spontaneously self-assembly form the capsid. You can have helical capsids, which are rod-shaped, and they form a continuous helix around the nucleic acid. You can also have an icosahedral capsid, which is these more geometric configurations. They can be made up of a single protein or several kinds of proteins. They have a 20-figure side with 12 evenly spaced corners. Helical nuclear capsid. The capsomeres are arranged like this with the nucleic acid in the inside. And this shows you a one with a cell membrane. Often they'll look like that if they don't have the cell membrane. Icosahedral capsids. This the adenovirus. You can see it has this very regular structure. It has these protein spikes. This causes, this is a respiratory virus. And you can see in these images here, the DNA on the inside, the capsid in the envelope, which is the cell membrane that the virus takes from the host cell. We talked already a little bit about bacteriophages. They're actually one of the most complex viruses that they infect the bacteria. They have this capsid head with the nucleic acid up here, collar, sheath, tail pins, tail fibers. And basically when they attach to the bacteria, they inject their genetic material down through here into the bacterial cell. Non-enveloped and enveloped viruses. Again, the non-enveloped, you can clearly see all the capsomeres that make up the capsid. Here in an envelope virus, the envelope is the thick membrane around it. And then you have the capsids and then the genetic material on the inside. The viral envelope, as I said before, is made up the membrane of the host, or it can be the cell membrane, or it can be the nuclear membrane. Regular membrane proteins get replaced with viral proteins. These spikes are usually glycoproteins, and they're necessary for attachment to host cells. Nucleic acids at the core of a virus, the genome, the full complement, as we said, the definition before, is the full complement of the genetic material. And that's the same for the virus. Keeping in mind that viruses either are DNA or RNA. The number of viral genes is small. So the COVID virus has just 15 genes. Some have more, some even have less. They possess only the genes necessary to invade hosts and redirect their activity to make new viruses. Now, the RNA viruses, they can either be positive sense or negative sense. And what that means is a positive sense RNA is the same thing as messenger RNA. So that virus, if it's a positive sense RNA, when it infects the host, the RNA can bind right to the ribosomes and start making viral proteins. Negative sense RNA cannot be translated. So the virus has to make positive sense RNA so that it can make the proteins and to make more of the negative sense RNA. So this is some viruses. Some examples: the smallpox virus is a double-stranded virus. Some reason it doesn't tell you here about the herpes virus. The parvovirus causes erythemia infectiosum. It's a skin condition. Single-stranded DNA virus. RNA viruses, poliovirus. It's a single-stranded RNA. It's a plus RNA, so it can go immediately to bind to the ribosomes. Influenza virus is single-stranded RNA, but it's a negative sense, so it's going to have to make the positive message RNA to translate the proteins. Rotavirus causes gastro upset. It's a double-stranded RNA viruses. Sorry, my dog's going, I don't know if you can hear that. Dog's going crazy because somebody's at the door. HIV is a single-stranded RNA. It has a unique enzyme called reverse transcriptase that the HIV makes DNA. The DNA incorporates into the host DNA and then either becomes senescent or later on makes new virus particles. Other substances in the viral particles, they do have enzymes, but some enzymes are coded for by the virus, but made by the host and then packaged into the virus. They might have reverse transcriptase. They have some polymerases, enzyme to make RNA, the reverse transcriptase that makes DNA from RNA, which is what the retroviruses have. Even some viruses can even pack some ribosomes, but they are the host ribosomes. They do not code for or make their own ribosomes. The retrovirus even borrows some host transfer RNA molecules, but again, it does not code for or make the transfer RNA molecules. So, true or false? Viruses can be seen with a light microscope. Is that true or false?
False.
That is false. Viral capsids are constructed from what subunits? What are the subunits that make up the virus capsid?
Aptamers.
That's correct. And the different structures, well, the icosahedral is, remember, that very geometric faced 20 different faces. The helicals, what it sounds like, it's a helical shape. Complex virus, actually, the bacteriophages are very complex viruses. List the 3 unique forms of viral nucleic acid. So, well, they can either be RNA, DNA, single-stranded or double-stranded. And that, to me, is, well, the unique forms would be if it's a negative RNA virus or double-stranded RNA virus or single-stranded DNA virus. Classification of viruses, informal classification system. They are classified based on whether they infect animals or plants, or bacteria, whether there are a DNA or an RNA virus, whether they're helical, icosahedral. The criteria for their formal classification refers to the structure, chemical composition, similarities in the genetic makeup. So the families are Viridae and the genre is virus. So some examples here. I'm just going to, these are not like viruses you have to memorize for the test. So like the measles virus, it's an animal virus. It belongs to the genus Morbillivirus in the family Paramoxoviridae. So these are just rabies. No, that's not rabies. That's the rubella virus. So these are just some examples of plant, animal, and one bacteriophage up here. Important human viruses that cause diseases. Ones you're probably quite familiar with, the smallpox is considered eradicated, and there are some that are still kept in laboratories in case we ever had an outbreak and had to make a vaccine. There's herpes 1 and 2. Herpes 1 causes the fever, blister, cold sores. Herpes 2, genital herpes. Of course, there's the chickenpox virus, and later in life, chickenpox incorporates into your DNA. That's why they say if you've had chickenpox, it's always in you. And so later in life, your immune system might not be working as well as you age. You can get shingles. Cytomegalovirus can cause, if I remember right, spontaneous abortions. This is a respiratory virus, and it's been modified, and so it's not infectious, but it has expressing the protein spikes of the COVID virus, and that's, I believe, the Johnson & Johnson virus. Hepatitis virus, hepatitis B. Some more viruses. Obviously, I wouldn't expect you to memorize all these. I'm pretty sure I referred to, I gave you a sheet that had the slides for the, what I consider most of the important slides for the test. These are viruses that used to not be much of a concern, but we now, West Nile virus has become more common in the US. It wouldn't surprise me with climate change that maybe some of these other viruses start becoming more common in the United States. Of course, the coronaviruses, which COVID-19 is a coronavirus. We're now finding that there can be quite, and this is also in the same family, the SARS virus. A few years ago, it in China, it caused an acute respiratory syndrome with a high fatality, but luckily did not become an epidemic or a pandemic. Ebola virus, be in close contact with people that have it, can be very infectious. There is a vaccine for the Ebola virus. Influenza A. There's another influenza virus. There's the mumps virus, the measles virus. You just need 2 vaccines. It's a very, the measles virus, its protein spikes are very stable. It doesn't change much. So you don't have to get an updated vaccine every year. Some other viruses less well known, but can cause problems. This is the retrovirus. You're already probably familiar with HIV, which is the human immune deficiency. There's another retrovirus, human T lymphotrophic virus that causes, it's actually a virus that causes leukemia, T cell leukemia in humans. Talk about modes of viral replication. So depending on their RNA or DNA, they have different ways they replicate. It's generally a 6. Make sure I have that person. It's generally 6 steps in their life cycle. We're also going to talk about something, cytopathic effects. When a virus infects a cell, it changes the cell, and we call those cytopathic effects. We're going to have some examples of persistent and transforming infections. Some viruses can transform a cell, mutate it, in other words, to cause it to become a cancer-causing transformation. And we've already talked about, but we can review again about the bacteriophage that causes lysogenic and lytic infection. We can review that again. That was an important way that bacteria, I'm sorry. Somebody had a question? That was one of the ways bacteria get genetic variability and can transmit virulence factors. So in animal viruses, the usual steps are all the same. There's the absorption. They have to attach to the cell. They have to get inside penetration. They have to lose that protein capsid. They have to carry out synthesis to make new viral proteins and to make new virus genetic material. That has to be assembled, and then they have to release from the cell, which they do either by bursting out, a lytic infection, or what we call budding, where they will leave the cell and take some cell membrane with them. So general features in the multiplication cycle of an RNA animal virus. So since they're RNA, especially the plus virus, what they can do is attach to the ribosomes and start carrying out protein synthesis. They have to make the capsomeres that make up protein coat. They have to make the viral spikes that then get embedded on the host membrane. They have to replicate their genetic material. And then they have to be released. So this is what I mean by budding. This is an envelope virus. So once the capsid is assembled and the genetic materials on the inside, it can go to the membrane and take this membrane, piece of this membrane with it, with the protein spikes that the virus had synthesized. General features in the multiplication cycle of DNA viruses. So DNA viruses, it's a little more complicated. RNA viruses in general is a general principle, replicate in the cytoplasm. To carry out DNA replication, the DNA virus has to get its genetic material into the host nucleus. And to replicate, so they're out here in the cytoplasm, they get their DNA into the host nucleus to make more viral DNA and to transcribe the viral RNA that'll then attach to the host ribosomes and make viral proteins. And to make things more complicated, the viral proteins have to come back into the nucleus and assemble. And it is there that the viral double-stranded DNA, in this case, is incorporated inside that assembled capsid. And then it has to leave the cell. So if you have a double-stranded DNA virus, it has to go through this. If it's just a single-stranded plus DNA virus, it has to make, at some point, a double-stranded DNA, so it can make more of the viral DNA and also carry out transcription to make the message RNA to make the virus proteins. The absorption process begins when the virus encounters a host cell and absorbs specifically to receptors on the cell membrane. So like the influenza virus has a hemagglutinin and neuraminidase spike. The one spike, the hemagglutinin spike, the H spike is to attach to the respiratory epithelia, and the neuraminidase spike is an actual enzyme to allow it to penetrate. So absorption to attach. So those proteins are necessary to attach to the host cell membrane. So that's adsorption. Absorb is to soak in like a paper towel. So just to distinguish, hope I'm saying that right. Adsorb is to attach. Host range of a virus. A virus can invade a host cell only by making an exact fit with a specific host molecule. So they have a restricted host range. For example, hepatitis B only infects liver cells of humans. Mildly restrictive hosts, so the poliovirus can infect both intestinal and nerve cells. Some viruses are broad range and can infect various cell types. So that's one of the problems with the COVID virus. It attaches to something called an ACE receptor. If you've heard of ACE inhibitors, they're involved in regulating blood pressure. And you have a lot of your different areas in your body has the ACE receptor on it, the endothelia of your blood vessels, liver, kidney, brain, heart, along with your lungs. So the COVID has a lot of places that it can bind to. So endocytosis, the normal process that cells use to bring things into the cell, and the viruses take advantage, trick the cell into bringing the virus in by endocytosis. The cell allows it doesn't allow, but it attaches to the cell membrane, and the membrane then enfolds and encloses the virus, allowing it to enter. Enzymes in the vacuole of the host cell dissolve the envelope and the capsid, but then that releases the virus particles into the cytoplasm, allowing it to begin its replication. Replication and protein production. Viral nucleic acid takes control of the host synthetic and metabolic machinery. Mechanism varies depending on whether the virus is a DNA or an RNA virus. As I said before, RNA viruses generally replicate in the cytoplasm. DNA viruses replicate in the nucleus. So do you have any idea like how many virus particles can be released from a single cell? So it's called the burst number, though we're not going over that, but it's called the burst number, how many virus particles could be released by a host cell. In the HIV virus, in a single T cell that they infect, it can release 50,000 virus particles. And so obviously lots of cells are getting infected, and in the millions of viruses are getting produced. And like in the case of COVID, every time that there's a new virus particle synthesized, it has a chance of mutating. So the mechanism depends on whether a virus is DNA or RNA. We already mentioned this, but in general, the RNA viruses replicate in cytoplasm. DNA have to replicate in the nucleus. The nucleus, picture of a nucleus of eukaryotic cells containing hundreds of the adenovirus virions. These are all the virus particles inside the nucleus of this cell captured in this electron micrograph. The release of mature virus. So envelope viruses are liberated by budding or exocytosis. So exocytosis is also something that naturally occurs in cells to release things that the cell makes into the environment. When the cell makes something, it gets packaged and then goes to the membrane, and the membrane pinches off and releases it into the outside of the cell. While viruses have taken advantage of that. So in the new virus, it's once it has the nucleic acid in the capsid, the whole viral nucleocapsid. For an envelope virus, it has to bind on the inside of the cell membrane, which is now has protein spikes, the viral protein spikes, and it buds off. It's another picture of viruses budding off of a cell. Viruses can be shedded gradually without the destruction of the cell. So the viruses, when they infect a cell, even if they're just budding off, not killing the cell, they can cause cytopathic effects. This is the damage that can actually be seen in a microscope. Cells can be disoriented, undergo major change in shape or size, or develop an intracellular damage. They can have inclusion bodies, which are compact masses of viruses, or damaged cellular organelles in the nucleus or the cytoplasm. They can form syncytiae, which is they can cause cells to fuse together into a large mass of cells containing multiple nuclei. I forget, there's one respiratory virus that does that, that tends to infect children, causes a bronchitis, and I forget that virus. So this is, that shows you an example of that. This is a normal cell, and this is one where the virus has caused multiple cells to fuse together. These are some examples of inclusion bodies inside a cell. So different cytopathic effects. Smallpox, the cells round up, inclusions appear in the cytoplasm. Herpes, the cells fuse to form these multinucleated nuclear inclusions. Adenovirus causes cells to clump. Poliovirus, though no inclusions, it just lyses the cell. Influenza, when it infects, causes cells to round up but no inclusion. Rabies causes some characteristic cytoplasmic inclusions. Measles also causes the fusion and the formation of these multinucleated cells. Persistent infections, sometimes a carrier relationship can develop in someone, and cells harbor the virus, but they are not immediately lysed. So you get in humans a provirus, which is viral DNA incorporated to the DNA of the host. That's what the HIV virus does, and the chickenpox virus. You can have a chronic latent state, a periodic activation after periodic of inactivity of the virus, which is what happens also with the AIDS virus and herpes virus. You can get infected and it can go into a latent period, and then some event causes it to come back out, and that's when you get like cold sores on your lips. It's the herpes virus. Now, viruses in cancer. Experts think that up to 13% of human cancers are caused by viruses. We call these oncogenic viruses. They lead to transformation. Virus carries genes that directly cause cancer. Virus produces proteins that induce a loss of growth regulation in the cell. So the genes that the virus has are called oncogenes. These viral oncogenes can cause changes and transformation of the host cell DNA. Another example here is retrovirus, the one that causes T cell lymphoma. It's like the AIDS virus. It has to have reverse transcriptase to make viral DNA. It has cellular oncogenes, and it becomes a pro-virus state inside the host cell. Can synthesize these oncogenic proteins that can cause transformation and lead to disrupting normal cell growth, which is tightly regulated, leads uncontrolled cell growth, which is basically what a tumor is. Okay, viruses that infect bacteria. We talked before about these. Do you remember what virus bacteriophage infection of bacteria was called? When we talked about that, the 3 ways that bacteria get genetic variability. It's called transduction. It was discovered in 1915, so obviously this is even before electron microscopes. Bacteriophages have the ability to parasitize every known bacterial species. This is even occurring among the bacteria in your gut. Often make the bacteria infect more pathogenic. So we said this was one of the ways the bacteria can acquire new virulence factors, is through transduction. So one of the bacteriophages, there's what we'll call T even, there's T2, T4. They're one of the most widely studied. They go through similar stages as an animal virus. When they infect, what happens is they bind to the cell wall of the bacteria. They have this tube. Their genetic material goes into the cell, so this part of the virus doesn't enter the cell, only their genetic material. We talked about the lytic cycle of these bacteriophages before. So they can take 2 courses, either lytic in the lysogenic stage, they become a latent prophage. Remember, in humans, it's called a provirus. In bacteria, it's a prophage. So the bacteriophage DNA is in the viral DNA is incorporated into the bacteria DNA, and it replicates right along with the bacteria. The other cycle is the lytic cycle. The virus puts its DNA into the host cell and then starts making more virus genetic material and virus proteins. They assemble, they build up in the host cell, and then eventually they break out of the host cell, and this is lysis. This is the lytic cycle. So it just leads to infection and destruction of the cell. The lysogenic cycle, they can go through a period of latency without making new virus particles. But eventually something can induce them to enter the lysogenic cycle. This shows electron micrograph, giving you an idea how many viruses can be either attached or breaking out of a host cell. You can see the entire cell is pretty much dominated by new virus particles. Bacteriophage life cycle, the lytic phase or lytic cycle, the life cycle of the bacteriophage ends in destruction of the bacterial cell. The lysogenic cycle, the bacteriophage becomes incorporated into the host cell as a provirus. I mean, as a, I don't want to get my terms mixed up, as a prophage, sorry. Provirus refers to virus DNA being incorporated into a human cell. That's a provirus. Prophage refers to the bacteriophage. So lysogeny, it's when it goes latent. We call those temperate phages. They have the ability to undergo absorption and penetration, but they do not immediately go into replication or release. During the latent period, they are a prophage. They're in inactive state. The phage DNA is inserted to the host chromosome. And then some event happens, an induction event activates the prophage to start making new viruses, and then it enters the lytic cycle and breaks out of the cell. And we've mentioned before the problem in human disease and transduction is these viruses can pick up from the bacterial chromosome toxins or enzymes that cause pathology in humans. We call this lysogenic conversion, the acquisition of new trait from a temperate phage. So some examples of diseases caused by lysogenic conversion: the diphtheria toxin, the bacteria have, the cholera toxin, and the botulinum toxin. So without lysogenic conversion, these bacteria that have these, the Clostridium botulinum, would not have the botulinum toxin without lysogenic conversion. Let me see, I think we have 71 slides. So what I'd like to do is stop here for a minute and call the names of people I did not see at the beginning, starting with Osman. Are you here? Odeneo, I'm sorry if I'm saying that wrong. Gaynor, last name Gaynor.
I'm here.
Okay, thank you.
I'm here.
I got you. Shamir Johnson, are you here? Jeremy Linton. Nikita Starks, are you here? Nikita Starks. Okay. Shamir Johnson, not here. And last Osman, B-A-H, last name B-A-H, you're not here? I mean, are you here? Okay, not here. Okay, just to remind everybody, next week is spring break. There's no class next Tuesday. April the 6th, the first Tuesday of April, we have exam 3. And April will be the last of our lectures, mostly on immunology. And May the 4th is exam 4, followed by May the 11th, the final. May the 15th is the last day that you can do the McGraw-Hill online assignments. So let me come back just a couple minutes and we'll finish this lecture. I think think I actually skipped ahead. Where were we? We're somewhere in the 50s. We have about 25 or so slides. Let me get back to where we were. I think we're on here. We're going to talk about cultivating viruses. So I'll be back in a minute. We have about 20-25 slides left. Okay, so let's continue. So viruses have to be cultivated in cell culture. We'll talk about 3 principles of cultivating viruses and ways that they are cultivated. In vivo means in life, a viral cultivation in lab animals or embryonic bird tissue. In vitro means outside of life, so this is a viral cultivation in a cell or in a tissue culture. The purpose of viral cultivation to isolate and identify viruses in a clinical prepare viruses for vaccines. When you're using a whole virus, we now have a new vaccine technique that was developed because of the pandemic. It's using the actual genetic material of the virus. Scientists have known for a long time this was something possible, and we saw it developed in rapid time, but we also still use the other techniques. Cultivating viruses in a lab for research on their structure, the multiplication cycles, their genetics, and the effects on host cells, the cytopathic effects that we talked about. Specially bred strains of animals can be used to cultivate viruses. White mice, rats, hamsters, excuse me, guinea pigs, and rabbits. Injection sites for viral exposure for these animals would be the brain, the blood, muscle, body cavity, the skin, or the foot pads of the animals. Eggs was one of the first. There's a series of, there's the cavity inside, there's the ambion, there's the yolk sac. All these can be sites to cultivate viruses in an egg. An egg is basically a sterile environment, so you can inject through the eggshell into the different areas of the egg to grow viruses. The embryonic development occurs in a protective shell. It's an intact self-supporting unit with its own sterile environment and its own nutrients, and furnishes several embryonic tissues that can support virus multiplication. Culture system or tissue cultures in vitro. It's outside of life, not in an animal. A simple and effective way to grow populations of isolated animal cells in culture dishes. Most viruses are propagated through cell culture, obviously very easy to work with. Most of a virologist's work involves developing and maintaining these cultures. Oh, didn't mean to do that. So these are some examples of cells and the susceptibility of a host to a virus. You can see the virus particle, the cytopathic effects caused by the viruses. Freshly isolated animal tissue can be then taken and placed in a growth media for a tissue culture. The cells undergo mitotic division, producing a monolayer on the surface of the plate. They retain characteristics of the original tissue from which they were derived. They can have altered chromosome numbers. After a while, they grow rapidly. You can see changes in their morphology. You can continuously subculture them if they're given fresh media. Some cell cultures that I've worked with humans, you can only culture them for a limited amount of time. If you take human cancer cells and culture them, they can basically grow indefinitely, though they can change over time. The degeneration and lysis of infected cells, you can see these plaques formed, clear, well-defined patches from the virus infection. You can see cytopathic effects. When viruses are released from infected cells, they radiate out to surrounding cells and then infect them. Infection spreads gradually and symmetrically from the original point of the infection in a cell culture. So the primary purposes of viral cultivation, there are several purposes. We can use the culturing to make viruses for vaccines. We can study the effects of the virus on cell cultures, the cytopathic effects. Influenza viruses have been traditionally cultured where? What do they usually ask you when you get a influenza vaccine? They usually ask you if you have an allergy to eggs. Describe the difference between primary and continuous cell lines. They didn't mention that in the slide, but primary cultures, when you take the cells directly out of an animal or possibly a specimen from a person. Continuous cell lines, you just keep culturing them. You see that a lot with cancer cells. True or false, the term plaque is used only to describe the effects of a bacteriophage on a cell culture. Other cells, other viruses can cause plaques. So other non-cellular infectious agents, these are, besides viruses, these are unusual, not very common. We'll talk about several infectious agents. One of the diseases is spongiform encephalopathy, which is called mad cow disease. It's a chronic persistent disease in humans and animals. brain tissue removed from affected animals resemble a sponge. And it's caused not by a virus, not by bacteria, but by a protein called prions. And these distinct protein fibrils are deposited in brain tissue. They have some weird ability to self-replicate. We do have normal prion proteins in our brain, the prions that cause mad cow disease or abnormal versions. Another disease, Creutzfeldt-Jakob disease, afflicts the central nervous system in humans, causes gradual degeneration and death. It's transmissible by an unknown mechanism. Several animals are also victims of the disease, causes scabies in sheep and mink, elk, and the bovine spongiform encephalopathy in cows. Prion infections, the exact mode of infections unknown. Protein composition of prions has revolutionized ideas of what we can consider something as an infectious agent. Questions about how prions replicate when they have no nucleic acid. So there's a lot we don't know about them. We also have something called satellite viruses. Satellite viruses can't replicate on their own. They're dependent on other viruses for replication. There's one called an adeno-associated virus. It has to have an adenovirus. It was originally thought it can only replicate in cells infected with adenovirus. They have now been found to infect cells infected with other viruses or have had their DNA disrupted by other means. Viroids are viruses-like agent that parasitizes plants. They are 1/10 the size of an average virus. They are only composed of naked RNA, so they do not have a viral capsid or any other type of coating, but they are in plants. They are significant pathogens in economically important plants, in tomatoes, potatoes, cucumbers, citrus fruits, and chrysanthemums. So, prions, what are prions composed of? DNA, RNA, protein, or polysaccharide?
Protein.
They're made of protein. Satellite viruses require a what in order to replicate? They require another virus in order to replicate. And then we have, already did that, but we have viroids, which are just naked pieces of genetic material that can infect plants. So I think this is the last section, viruses in human health. Analyze the relative importance of virus in human infection. Discuss the primary reason that antiviral drugs are more difficult to design than antibacterial drugs. It's impossible to measure the number of viral infections worldwide. Most common cause of acute infections that do not result in hospitalization, colds, chickenpox, influenza, herpes, warts, they're all caused by viruses. Viral infections that only occur in certain regions, such as dengue fever, Rift Valley fever, and yellow fever. Some viruses have very high mortality rates, such as rabies and Ebola. And the one coronavirus, the SARS virus, also has a very high mortality. Other virus infections can result in long-term disability, such as polio and neonatal rubella. Viruses can mutate at a rapid rate. In general, RNA viruses are more mutagenic than DNA. It's difficult to design therapies against viruses. Scientists focus on developing vaccines since so few antiviral drugs are available. and antibiotics, of course, ineffective against the virus. Though I think the current, Ebola, first the Ebola pandemic, and then now the COVID pandemic, there is a lot more interest now, it seems to be, in developing antivirals. And especially considering how widespread the infections are and how difficult it's been to contain them. A lot of countries have little or no access currently to vaccines. I was reading about Russia. I think less than 10% of the population has been vaccinated in Russia, even though they've supposedly developed several vaccines. In this country, I think we're now close to 50, not 50, I forgot, it's millions, maybe we're close to 50 million people. I think it's in the 40s of people have been vaccinated. We have 3 good vaccines, a possible 4th one. We are buying, the government's buying extra doses of these vaccines to give to Mexico and Canada. Because obviously we share a border with both these countries. There's a lot of movement across the borders, and it's very important to get them vaccinated to protect our people in the US. Interferon is a naturally occurring cell product, and it's when a cell gets infected with a virus, it'll release interferon that'll signal other cells to make antiviral proteins. We can now use DNA technology to make interferon. Viral infections are more common than bacterial infections. What do you think, true or false? I would think that's true, but it's hard to determine. It's hard to get accurate numbers. Even in this country, for example, influenza is not considered a reportable disease, but a lot of one of the problems is a lot of people get influenza and they just stay home and they get over it, or they don't go to the hospital till they're really sick, and then sometimes they get a secondary bacterial infection, or if they have other health issues, they might die of that, which is then how it's reported.
I would think also that the viral infections often come back, like things like herpes and Epstein-Barr virus, and that they stay with you a long time.
Yes, some of those, if you've had mononucleosis, one of the problems is some people can develop a lymphoma from the mononucleosis infection caused by Epstein-Barr virus. Obviously, the measles, the chicken pox, once you've been infected, you have that for the rest of your life in you. And the same thing for the herpes virus, I think you were mentioning. True or false, viral mutation rates are low and viruses are easy to track in a population.
False.
Yeah, it's false. Viruses can mutate very readily. We're seeing that happen before our ideas, why we're still trying to get people to wear masks and social distance. Even though more and more people are vaccinated, we are not near herd immunity. And it's not easy to track viruses in the population, though we do have testing now, but that depends on doing the test and people's willingness to take the test. Where I work, we do a pooled RNA test. I can get tested every week. And occasionally we've had somebody show up positive. So far, people that have been positive were people that contacted the COVID outside of work. It's only been a very few number of people, luckily. Most doctors and scientists often rely on blank to treat or prevent viral infections.
I don't know if it's antibiotics or vaccines.
Vaccine.
Antibiotics.
The antiviral drugs. So I think I mentioned before, back before Thanksgiving of last year, I got the flu. I don't know how I got it. And they put me on Tamiflu, which seems to work pretty good. Okay, so that's the end of that. If anybody wants, I have the last two lectures. I'm going to make up quizzes for, and I want to just stop the share for a second. And